Okay, good morning, everyone. So just going to give you a little bit of an overview of Dovetail Genomics. So I think we met yesterday, most people. So my name's Miles Collier, based out of Cambridge in the UK. I put my email address there, but I've got some cards if anyone wants them for later on. And also introducing Lily, who is hiding behind the mask there, who is our Director of Lab Operations. So just giving you a little bit of an overview as to Cantata Bio, Dovetail Genomics. And really, we've been around for a little while now. We started out in 2013, so around about 9 years ago, by Ed Green out of University of Santa Cruz. And really what he wanted to do was establish Dovetail as the leader in genome assembly. I think that's where Lily first started with the company, knowing Ed from all those years ago. So we've got that continuity as well. ArcBio is our sister company, and that was formed a little bit later. And we're both companies are part of Eden Rock Sciences, which was founded in 2016 under the direction of Ed Schulak. So Dovetail, really, we started producing products in 2018, and on the ArcBio side of things, they started in 2021, and then we became a merged company in 2022 under the name Cantata Bio. We've kind of reverted back to Dovetail Genomics for two reasons. One, it's a much better logo, and two, nobody can say Cantata Bio, especially in the US. So it's much better to be known as Dovetail, and I think we're happier that way. The ArcBio side of things is really going to come online later this year. So watch your space if you're in the microbial world as well. So a little bit of what we do that is slightly different. And this was a quote from our R&D director, Lisa Munding, is that Cantata Bio believes that HiC libraries are better when we don't introduce the bias of restriction enzyme fragmentation. So all of our products will not use restriction enzymes. So I know you're all here to learn HiC, perhaps a little bit of a background from it. So it's increasingly well cited. It was developed in 2009, Lieberman et al. In Science, and really was there to investigate the 3D structure of chromatin. As a method, HiC is there to map chromatin contacts across the genome using next-gen sequencing, and it looks at interactions between areas that can be either quite close together or thousands of bases or hundreds of thousands of bases apart as well. Okay, and I think that the important thing is that any interactions that are seen through HiC, you wouldn't necessarily see on the linear genome. Okay, so a little bit about our expertise, and some of you in the room will have used OmniC, sort of the sister product. This is a product that uses DNase I as our fragmentation enzyme. That's to digest the chromatin, and then it goes into the proximity ligation assay. OmniC is kind of the throw-all, capture-all as a product. It works on pretty much everything. So it's definitely sort of our flagship. But we're moving into some of the other things. The workflow will be the same for every HiC experiment. So cross-link, fragment, ligate, reverse the cross-links, and sequence. So the workflow is the same. The real difference between this and any HiC is the fragmentation steps. By using DNase I rather than restriction enzymes, what we have is far more uniform fragments. And because we've got more uniform fragments, that gives more uniform coverage. We'll go into this. So in terms of diploid assembly, with HiC, what we can do is get fully phased. So you can get mum, dad, in terms of chromosome structure, which is something that you wouldn't get again through normal sequencing. Kind of touched on this already, but if you look at the distribution with OmniC, it's far more uniform, so it will give that improved coverage. And final one there really is that OmniC does behave like shotgun sequencing as well, so you don't have to run a normal genome analysis plus OmniC. So here I'm going to introduce AssemblyLink, which I'm going to call the jewel in our crown. It's something that we have literally just launched, as shown by this press release that came out, 10 days ago. So you're one of the first in the world to really hear about this. It's something that we're very excited about. So AssemblyLink, really what we're trying to do here is we offer a very fast workflow. As Morten said yesterday, this is the only one that you're going to be able to go through to completed libraries in this week. So it's going to be much quicker. One of the nice things that we've seen is that it does take low input samples as standard. So at the moment, we've seen down to around about 5 milligrams of tissues, which I think will cover a lot of things. It's certainly around about a third to a quarter of what OmniC typically works with. Same methods. It's a shotgun-like sequence coverage. It supports scaffolding, and it gives us other things like the genotyping and haplotype phasing that we would expect from HiC. Protocol or the workflow, I told you it's all going to look very similar, and sure enough, it does. So you start off with your sample. We fix. We've got the two cross-linkers here that we have in some of our other products. Digest, ligate, and then go on to sequence. But the real advantage is all of that is done in a single day. Kind of hammering the point home there. So how does it work quick? I think this is one thing that, to me, it was much easier once you had this visualization. So in terms of sample prep, it's going to be the same for whatever you do in terms of HiC. It's this proximity ligation that is very much shorter. Okay, and that's how we're able to get it down to the single day. At the moment, we have validated a number of different tissues, a number of different sample types. So mammalian cells, we know it's going to work. And I think that's one of the sample types we're going to do here. Mammalian blood, we know is going to work. Animal tissues, we've validated things like the mammalian, we've validated fish. Some insects we know that are going to work as well from what we've tested. What we haven't yet validated is nucleated blood. We're working on plant, algae and fungi, and some of the nasty samples you guys are throwing at us. We're working on to try and get as close to sequenced as possible. Watch this space. So, real features and benefits here. The rapid workflow. It does mean that you can come in the morning. If you're prepared to have a long day, you will finish before you go home. So that way is nice. It's simple to use. It's really there for whoever has got a reasonable amount of molecular biology training, which is why I'm not doing it at the moment, but you guys are. The workflow is easy. We do know that people are looking at automating at least parts of the workflow as well, which makes life a lot easier. The low sample input amount we think is going to be really useful. I know sometimes you'll come up with organisms that aren't 5 milligrams, but we're working on as low a sample input as possible. And the data so far that we've seen has been beautiful. So again, looking forward to seeing how this works. Data analysis, we do have our Read the Docs page, as we do for all of our products, which really take you through the whole workflow. The link's there. Again, if you want, we can send you these details and you can follow it that way. We've used our own high-rise software, which we know works, but we've also used tools like Yaz. So I think what many of you are going to be using has already been covered. Again, the one thing that we think is important is that it's uniform coverage. So the size distribution should be really the key here, and that's something that we found with using AssemblyLink. So you should get good even coverage across the genome. Yeah.
The skewed coverage, different coverage, is it depending on the GC bias or the chromatin structure, or why as compared to multi and single R?
That's a good question. Lily, do you know that one?
Sorry, say again, I can't.
Is the coverage due to GC bias or anything like that? Why is it higher?
Oh, I mean, the coverage is, excuse me, it's just about where you can sequence the ideas with restriction enzymes. You know, I can't remember which one that's using, like whether it's a single or
Both. We've got a single and multi.
Yeah, right. So
Yes.
it's sequence dependent.
Correct.
Yeah, okay, I got it. Thank you.
Okay. So again, the haplotype resolved assemblies is something that we're able to do as with all of the HiC applications. Testimonials, we'll kind of skip over some of these because someone's in the room who's already given it. But I think one of the things that's nice across all of the testimonials is that people are excited about this. This is certainly going to make life a bit easier, a bit quicker. Some labs have already had early access and tried it. Others of you are just looking for a faster, easier, more robust way of doing the HiC. So definitely the promise of AssemblyLink is exciting. So we've got a few of those. Again, happy to show you those, or go on to the press release and you can read them at your leisure. Ordering information, for those of you who are interested, drop me a line, drop me an email, and we can get a quote out to you. It is available now. You can buy it now. So it's not only hot off the press, but it's available to be in your lab in a few days' time. So just a bit of a recap on what we say. AssemblyLink offers a rapid workflow, supports low input of samples, uniform shotgun-like sequencing. It supports all of your standard workflows and is available for genotyping and haplotype phasing. And with that, just got a couple of other slides, what you would need to supply yourself. I think this is kind of useful for you to know. So you'll have it all in your lab, especially if you've been doing HiC, but these are the things that we ask for. Yes.
No, sorry, you can continue.
No.
Have you taken a little automation? Can you say that?
We know that people are looking to automate. Mike is one of them who I think has put a HiC protocol on the Agilent Bravo, and I think he's looking at the star as well, Hamilton star.
Yeah, we are in the process to use, but you know, it's a difficult pipeline.
It is. Yeah, and I think some of the extraction you'll never automate.
Yeah.
But because there are fewer steps, we think it is automatable from a certain point onwards. So I've spoken to a few companies who are interested in looking at automating, but we know that there are some of you out there who are looking to automate as well, especially when you've got the pipelines of sort of 96 samples going through at one time.
I don't know anything about Dovetail, but you have the PCR cycles, I suppose.
We've got a few cycles, so a few heat and cooling cycles, yeah.
And that doesn't introduce any problems in the end, because sometimes we have to make a realization because it doesn't work very well.
Yeah.
Do you have that?
Again, Lily, is that something you can answer on PCR?
say reapplication.
We don't have a lot of material to sequence it, like we need 2.8 to go to the Illumina, for example. And sometimes even if we follow the concentration that we know the concentration and we are going to do 12 cycles and that it doesn't work. And so we have to introduce a little more, maybe 4. And we are worried that we are amplifying something that in the end, we don't need it or is just.
Yeah, I mean, in our experience, and maybe Brian can speak to it. If you don't get very much library yield, it means you didn't have enough material to begin with, so you can do more cycles, but you're not gonna get more information, so usually that's a start over.
Yeah, sometimes it works,
Yeah.
but not always.
But yeah, don't you think you over sequence afterwards when you do deep sequencing?
Yeah.
You sequence quite a lot and you lose a lot of rates because of the duration. Yeah.
But this happens especially in the ultra low.
Mm.
It's very low. So, but we try to not do a lot of cycles because it's not good, right? But most of the time we have to do a little more. Yeah, we can discuss maybe on Thursday. I think with the data here and everything, we can see better.
So the number of physical cycles with AssemblyLink is how many?
I believe it's 12.
Is below 12, no?
Yeah, 12.
Oh, so.
In the, on the border.
Yep,
Mm.
thank you. So just a quick overview of the workflow. As I say, it is single day. There are stop points that you can do. There are QC checkpoints as well that you can do. So, I think here you'll be doing it over 2 days just to get yourself a feel to it. But once you're up and running, it can be done in that single day as well. But yeah, it's nice to have safe stop points as well. In terms of sequencing, how many kits that you'll need, it's very similar to OmniC, so we'll do it in 3 gig iterations. If you want the slides, we can send these out. It's not a problem. Yeah,
The big presentation is recorded on Zoom, so you'll get everything, I promise.
absolutely.
Yes.
Sorry, so the coverage suggested is lower than in the OmniC?
No, it's similar.
Is the same?
Yeah.
Because I remember was 50, the Excel, the suggested, maybe for the Lima, no?
Yeah, I think different groups, right, like EVP, and also other people, sorry, my brain is half working with this, but normally do 60X.
Yeah.
Yeah.
BGE
Well,
50X.
Okay.
Personally, I think the reason is because as you lose a lot of valid pairs, it's called valid pairs.
Yeah.
I mean, to maximize the number of valid pairs, you increase the coverage. So if you expect a good library, maybe to end up having 60% of valid pairs, that would be super good.
Yeah.
Then, I don't know, from 300 million bit pairs, you will end up having 180 million pairs that are valid.
Yeah.
And this comes to be 60 million per gigabase of genome in a map. When I see the contact maps for that density of contacts, it's quite good. The scaffolding works well, and the correlation is easy. But then when you drop this down, you start having problems with the scaffolding or correlation. So that's the reason why the people increase that. In human, probably with 30x is very good. Yeah. I mean, that's my interpretation.
Yeah, no, it's true. Looking at somebody say that they use 60X routinely because some samples are really 60X, so why try to estimate every time if it's in sequence? Maybe you have more data than you need, but.
Yeah, but it's good. It's good anyway. It's not strong to the news.
Thank you. It's good discussions. Thank you.
Sorry about that.
No, good. Thanks. I think that's the end of that presentation. So yeah, thank you for your time. Thank you for your questions. Anything more? Thank you. Anything more? I'm around for the rest of the day, and Lily, as your expert tutor, is around for the rest of the week. So thank you very much.
I have another question.
Yes.
When you tested this in different tissues, for example, in a human, you tested different tissues for doing the AssemblyLink protocol.
Yes,
And you saw difference between the tissues like a reaching fiber, like the muscle, brain.
active spleen.
They still being the same as before for only C, or they are lower?
No, the input, I mean, or.
I mean, the results you get from doing the library.
Yeah.
And
So the results we get are the same, but in general, it's the way the AssemblyLink works, we need less input.
you need less even with
Less,
with these
yeah.
soft tissues.
Exactly, yeah. Because it's a PM5, like, you kind of want to be in a good range, right, for the input. So less is also.
And you tested this with mollusks, for example, that are reaching glycogen.
Yes, we
Or-
can talk more about
Or
that.
corals or things like that, because
Yeah.
I mean, if it in human tissues seems to be
Easy.
more stable
Yeah.
across tissues, no?
Yes.
I would expect that we have some hope with this challenging samples.
Yeah, that is, we can take it on Thursday because there will be, many people with mollusk problems.
But
In the room.
to answer your question, we developed the assay using cell lines and mammalian tissues because they're easier, so. And we know what the baseline is, and then we can move on to see cucumbers or.
Thank
I have a question. So, because the coverage is so uniform, so sometimes when people do sequencing on Oxford Nanopore, it's the genome skimming with Illumina is recommended in case if you need to correct for the indels and so on. Can you use AssemblyLink data to correct the nanopore reads throughout the entire genome?
It's a good question. We don't tend to do an awful lot with nanopore at the moment, so I couldn't say for certain, but we know people are kind of picking up and using it. It's just they're not really coming our way.
It's something I thought about that, but the problem I see is that with parent data, you have parent information, and then the mappings are probably more robust to correct a possible indel. With only see you will have single end. You have to treat it as single end because the distance between the two pairs, or what is called the insert only, I think, is super
It's too big,
It's abnormal, no?
Yeah.
But what we are doing now, for example, is we are pulling the high-fi reads, the Q20 reads of Oxford Nanopore and the OMC data or on the Illumina, sorry. We pull it to do the camera profile.
Okay.
And then when we estimate the UV, that's the accuracy of the sequence after sampling. Using both that kind of reads, we get similar estimates. And normally in a well-behaved genome, vertebrate, we get QVs that are over 47 and so on, both with Q20 and with parent. So at some point, I think we can even get rid of the polishing with nanopore.
Cool.
I think.
Yeah.
But still, it's nice to have both, because then there are species that is very hard to examine the genome when you have low coverage, because they have a huge genome, or they are tetraploid or things like that. So then pulling all the data. Well, sorry.
No,
We're talking over the coffee break.
And on Wednesday, because Fernando now he's mimicking like one of you guys, but as a matter of fact, he's a bioinformatician.
In disguise.
Of course,
So he infiltrated the lab, which is actually, I salute you, because not many bioinformaticians want to try it actually hands-on.
I have the help of.
I think we're definitely we're interested in seeing what the nanopore, because we're seeing it more and more. So yeah, it'd be good to hear some more.
All right.
Any other questions? Thank you.